Hello, this will be your first video of AS Bio entitled Science and the Scientific Method. So I know you guys have thought about this before, you've talked about it in formal contexts in class, but we're going to talk about what science actually is. Science, in a very general term, is the study of the natural world. And there are three broad types of science, and really this is types of science careers. So there's life science, which is the study of living systems and the way in which they interact. There's earth science, which is the study of earth systems and systems in space. And there's physical science, which is the study of matter and energy. So life sciences tend to be biology, anatomy and physiology, marine biology, and other similar studies. It is the study of life, the study of the body structure, and the way it works, the way it reacts in its environment. Just like marine biology is the study of marine life. It can even get more specific. There are different types of biological studies for almost any type of environment that you can think of. Okay, and so in earth science are disciplines like geology, marine geology, astronomy, etc. Geology is a study of land formations and the processes that form their composition and structure. It's a big part of understanding planetary science, other planets, how they are formed, and how we can study and understand more about them. Marine geology is the study of geology as affected by the marine environment. And astronomy is the study of space and the systems within the universe. So by no means is this even close to all of the earth sciences that there are. There are many. These are just a few examples. Okay. And within the physical sciences, so physical science studies are like chemistry and physics. Chemistry is the study of matter and the way it interacts in the environment. And physics is the study of energy and its ability to make and change matter. So physics really are the physical laws that govern how everything else works. And you guys have had introductions to most of these sciences. So science really is the process of studying systems. So that can be an anatomical system, an ecosystem, solar system, chemical systems, or physical systems. It's about understanding systems. Okay. A system is a group of structures, cycles, and processes that are related to each other and work together. And I mean, really, all systems are made of other systems. And so trying to understand what those systems are and how they interact is the process of science. Science begins with inquiry. Inquiry is asking questions about things that you see in the world. The answers are found by further observations and experimentation. And sometimes information can change because of new discoveries. When that happens, we have to reevaluate the science that we thought we knew, and that's the beautiful thing about science, right? Is that it's always a system that's reinventing itself or re-examining itself or reanalyzing. Okay, so this is this flow chart that kind of talks about how science works, and I love this flow chart. Usually this is where you would see a graphic that says the scientific method, and it would start with make an observation or make a hypothesis, and then do an experiment and analyze the results, and then either publish, accept your hypothesis, or start over, and it's sort of this linear process. But that never felt quite authentic. So this is a model that was developed by my colleague, Dr. Jessica Bean at Berkeley. And, I really, the second I saw this model, I really fell in love with it. So this is a nonlinear process about how science actually works. And so, just take a few minutes to really You pause the video, take a few minutes to really look at all of the components of this model. So so go ahead and pause it now, take a look at it. Then when you hit play again, we'll discuss what is actually in the model. Okay, hopefully you took a good look at the model. So if you start at the top, it says exploration and discovery. So I really love those two arrows that are coming into the side of this model that says new technology, practical problem, curiosity, personal motivation, serendipity, surprising observation. So this is usually where on a traditional linear flow chart it would say observation. So it's not always about an observation. Sometimes it's that a new technology is developed which creates a situation or a need or a product that is new to us, that needs to be studied and needs to be understood better. Sometimes it's serendipity, right? Luck. Something happens and it really sparks curiosity, or really you get this really good idea but needs to be tested. Okay, so that's where really we start. And then you move into this exploration and discovery, where this is where you're making observations, you're asking questions, you're finding inspiration, you're exploring the literature, you're sharing data and ideas. So moving through the flowchart, you move to gathering data, okay, interpreting data, and then those two bubbles on the sides, you have benefits and outcomes and community analysis and feedback. And so as you can see, these arrows, they're moving in and throughout this model constantly. And that is the beautiful part of this, is that is exactly how science is. It is not this linear thing. Like, it's not like you just start with a hypothesis and then you go through the steps, and if you go through every step, you'll end up with an answer at the end. Not how it works. And so we're going to spend a lot of time using this model throughout the year. I'm going to ask you many times, like, where are we in the process of science? Which place are we at? Are we at more than one place? Are we moving into more than one place at a time? So you're going to have paper copies of this and a PDF that is uploaded on Schoology. I highly recommend spending some time with it. Okay, so before science begins, a person makes a prediction, or which usually is an assertion with supporting evidence. Okay, and this evidence generally isn't formal evidence. Okay, it's not evidence based on studies that have been done necessarily or papers that you've read. Really, it's evidence that is observation, so it's anecdotal. Observations and prior knowledge gained from previous experiences allow a person to make an educated assertion, which is testable. Okay, and that's the key here, is that it has to be testable. If you're making an assertion about something that can't be tested, that doesn't mean that it's less valid or that it's not possible. What it means is that it can't be tested. And science is a process of testing. So if it's not testable, then it's not within the purview or within the realm of science. Okay, so this is known as a hypothesis, and an experiment allows a person to test the validity of a hypothesis. So if we're looking at the scientific method, we're going to think about that really complex model that we were just looking at, and we're going to pull away from that a little bit and try to take a more linear sort of path through that. Okay, so I'm not going to talk about every little bubble that we were looking at. I'm not going to really talk about serendipity or about community analysis and feedback. We're going to talk about that core linear process of the scientific method that you're already familiar with, which starts with a hypothesis and moves into asking a question. So here's a question. What is the effect of increasing light exposure time on plant growth? Okay, so to start, you're going to look up background information. And why would you do that? Why wouldn't you just jump right in and start trying to answer that question? And really, to learn the pertinent details about the system that you're researching is really, really important. One, that answer might already exist, and in the process of doing actual science, like you are a scientific researcher, you do not want to ask or try to answer a question that's already been answered. And if it's an answer if it's a question that's kind of been answered before, but not fully. You want to know all of those kind of answers. You want to know everything that is pertinent to your question so that you can set up the best experiment possible and have the least surprises. So, an example, plants grow as a result of storing excess glucose produced in photosynthesis as cellulose and other plant structures. Light, water, and nutrients are required to perform photosynthesis. Okay, this is something you need to know. If you're going to perform an experiment about photosynthesis, you want these details. Okay, and you're in these details. You're looking up this background information, and it's to make sure no one has already done it. So we said that already. Okay, so now as you're formulating a hypothesis, really in most cases you're formulating a null hypothesis and an alternate hypothesis. So a null is there is no effect of light exposure time on plant growth, okay? And your actual hypothesis or your alternate hypothesis is increased light exposure time will increase plant growth. Okay, so then you're going to design a controlled experiment to test your hypothesis. So first, you're going to start with your independent variable. You guys have, I'm kind of rushing through that. I'm not rushing through this, but I'm kind of flying through it because you guys have experience with all of this stuff. So your independent variable is a variable that you're manipulating in order to test your hypothesis. So in our photosynthesis example, exposing the plants to various lights and exposure times, okay, 1, 2, 3, 6, 9, and 12 hours is going to be your independent variable. It's the part you're manipulating, okay? And then your dependent variable is the measurable variable that changes as a result of manipulating the independent variable. So your example would be weighing the average biomass produced by the plants during the experiment, and you're doing that to see if your increased time changed the biomass of the plant. Okay, and your controlled or stabilized variables. These are variables which are maintained constantly so as to isolate the independent variable. And again, they're known as the constants. So an example of this would be the water, the type, the age, and size of a plant. You want to control all of those because let's say you did an experiment in which you didn't control the amount of water that a plant was getting. Some plants were getting a lot of water, some were getting moderate amount of water, and some were getting very little. And you also were manipulating the amount of light they were getting. Well, okay, in the end, plant A, it grew bigger. It has a lot of biomass, and you're like, yeah, I have good results. However, because you didn't control how much water you used with the plant, you can't say whether it was the water or the increased light that increased the biomass. So then you would have to start all over again. And so this is why we control variables. We control as many as we can think of, as many as we can, to really isolate that independent variable for testing. Okay, so you create control and experimental groups if it's applicable to the question that you're asking. So a control group are test subjects that will not be treated with an independent variable. So in our example, if you're testing the effective light exposure time on plant growth, the control plants in the control group will only receive light for a specific amount of time, not the varying amount of time that your test subjects are receiving. This is sort of like going back here, this is kind of like a secondary control to double make sure that any results that you see are because of the independent variable. Okay, and then, so in a pharmaceutical testing and a few other different types of tests, there are control groups which receive a placebo. And a placebo, an example of a placebo is like a pill that is identical to the experimental pill that is really made of sugar. And the point of that is to eliminate bias. So human test subjects, if they know they're taking medicine, they might report effects of the drug based on the knowledge of whether they received it or not. And it's not always that they're lying. Sometimes, the brain can be a very powerful thing. And when we know that we're receiving or not receiving something, it can have an effect on our physiology. Okay. And again, sometimes that's a person lying and saying that it's having an effect, and sometimes they really think it is. Okay, so this is an example of a control group. So the experimental group, these are the test subjects that will be treated by the independent variable. So in our experiment, it is the plants that are receiving the variable amounts of light. So then you design the procedure. Designing the procedure is incredibly complex. You have to make sure that you know where all the safety equipment is. You have to know all of the ethics that are involved. So specifically, if you're working with anything that's living, okay, especially people or animals, you have to go through a very extensive review process to get approval to do that kind of work. Okay, so you're going to gather materials and you're going to test the procedure and modify the procedure and materials if needed. And really, replicates are really important. Replicates during the experiment allow data to be averaged and make the results more accurate. So we generally call replicates trials, right? So we do more than one trial to make sure it wasn't just kind of like a fluke. Okay, so then if it's appropriate for your specific question, you would build a model. So a model is something that represents an object, event, or idea. Models can be used to communicate ideas, test predictions, and test safety. So there are three basic types of models. There are physical models. These are models that you can touch and see. There are computer models, models you can see but not touch. And there are idea models, models you cannot see or touch. Okay, so talking about physical models, so this is like an actual globe, right? It's something you can touch, that you can manipulate, you can see it and touch it. Also the solar system, so having models of the planets, okay? Computer models are models that you can see but not touch, so that's using various statistical or GIS programs to create models that help you to understand large data sets or trends and or actual physical representations of things like an atom. And then there are idea models, and these are things that you can't see or touch, and these are like equations. Okay, equations that can be used or models like evolution or natural selection. These are models that you don't see or touch. They're ideas that are used to explain complex events or series of events. Okay, and then you run the experiment. So during the experiment, be sure to observe and note anything unusual. And you really do this to account for anything that might cause an error in the experiment. So it's really important to try to take notes and to just based on your observations of what you see is occurring, because at the end you'll get all of this data, and sometimes you'll see this little blip in the data where you think, oh, that's unusual. Is that something significant or insignificant? And so the first place you would start is back in your notes. So you would really look back to say, oh, did anything weird happen right there? Okay. You make your observations, you collect your data. You can take qualitative observations or quantitative. So qualitative examples of that would be like, my plant is alive or dead, or it is flaccid, or it is febrile or fine. Okay, these are qualitative descriptions of what is happening. Or you can take quantitative observations, which is, my plant grew 3 centimeters per week, which calculations show me is a 54% increase over time. Okay, so what kinds of data do you have? So two things that you really want to think about, and we've talked about this and I've showed you this model in class before. You really want to think about your precision of your data and the accuracy of your data. So if you look at this model, it really is really a great model to show the difference between precision and accuracy. So in precision, data points that are closely grouped together are precise, but that does not mean that they are accurate. Okay, so if you look at letter B, okay, those 3 darts are super close together. That is high precision. Whoever is throwing those darts is very precise and able to really get the darts really close to each other each time, but they are not accurate, right? They are far from the target that they are actually trying to hit. Okay, so then in accuracy is when data points truly represent what you would expect or what they should represent in the system or experiment. So if you look at C, letter C on the model, you see that while the darts are sort of spread out around the bullseye there in the middle, all three of them are extremely close to their target. Okay, they all have high accuracy, but they have low precision because they are at different places on on the target. And so when you look at D, you see that all three of them are right in the center there of the target. So they have high accuracy. That's what you're trying to get to. That's where you want them to be. But they are also highly precise because they are all very close to each other. Okay, so you can see this is another model to show you on a graph. So if you look at the blue curve is a normal distribution of data, okay, like a bell curve. The green curve, it is precise, but it is inaccurate. So you're looking at it, and it is also showing, so if you look at the mean of that green curve, it is also similar to the mean of the blue curve, okay? But it's inaccurate because it's not accounting for, well, first of all, it's shooting up way higher than the normal distribution is, and it is not accounting for all the variance under that curve. So it is precise, but it is inaccurate. And if you look at the red curve is less precise, right? It has a further, wider distribution of variables and data, but it is more accurate. It's closer to that curve, to that normal curve. Okay, so then we analyze our data, and data should be organized on a table. Data can then be plotted on a graph to look for trends. So you can have tons of numbers in an Excel sheet, but it doesn't really help you until you graph it, okay? So I'm looking at a tomato plant response to light. Here's a beautiful table. It has a title. It's got your independent variables on there. It has all of the relevant data that you collected, okay, and which is your dependent variables. Okay, here is a graph showing that same data. Okay, so how much easier, so looking at this data table, you can pour through this thing, and maybe it would be a lot harder to see the trends here. You could get there. It would just take a lot more work, and it's not as apparent. But look at how beautiful that is. You can really see what's going on, what the trend is with this tomato plant. Okay, then you're drawing conclusions from your data. Okay, using critical thinking skills, you're going to discuss the meaning of the trends, noting any extremes in results and statistical significance of results. Sorry about that. So when you've gone through this entire process, it's time for you to submit your research for peer review and to report the results to the scientific community. So why? Why does anyone do this? Peer review ensures quality of research. It is also important to ensure published research is not biased by the researcher. So how would peer review ensure quality? So there are plenty of reasons why, but a few of them, when, and by peers, it's really a journal where other scientists read your work. And so in a field like science, there's always a level of competition for grant, right, to pay for your research. And so when other people are reading your work, I mean, honestly, they're reading it with a very critical eye. And when they do that, it helps to make sure that your methodology is sound, that your research hasn't already been done, and that your interpretations of your results are plausible. And so because of this, there's this sort of instant peer review feedback system that really helps to keep science a discipline that is growing and constantly analyzing itself. So it's also important to ensure published research is not biased by the researcher, and that's something that an individual researcher can't always tell, right? You can't always assess your own biases. So it's a really important part. If the results are hidden away, the research benefits nothing and no one. So when you are reporting your work in journals and at scientific conferences and in internet abstracts, you are sharing your work with other scientists and hopefully with broader communities where the benefit of your work can be felt. So based on the conclusions of an experiment, you can make inferences about similar scenarios. So observations and results from many investigations may lead to the formation of a scientific theory. So here's the definition of a theory that I absolutely need you to understand and to know. A theory is a model, okay? It's a model that explains diverse patterns that are seen repeatedly in the natural world. So that's it, okay? It's a model. And the reason why it's a model is because it's an explanation that is meant to sort of be this umbrella over many, many different events, okay, or many different systems that are working together. A theory is not a law. A scientific law you can think of that as like a rule that describes a pattern in nature. So where a scientific theory is a model that explains diverse patterns and systems, a scientific law is a rule that describes a pattern. Okay? So gravity is a theory and a law. Okay? The law of gravity states that a dropped object will fall back to Earth. The theory of gravity attempts to explain why and how gravity exists in all of the ways that we can measure and understand it. Okay? So this is not a theory or a law. Okay? Ha ha ha, not a funny joke. Okay, so science answers questions like, why do the seasons change? What is the effect of salt water on plant growth? Why do I need oxygen to survive? Science does not answer questions like, who should I vote for the presidential election? What is the meaning of the poem? Is interrupting someone rude? Okay, so what does technology have to do with science, or how does science use technology and vice versa? The application of scientific knowledge to be useful in our everyday lives is done through technology. Okay, so all of these pictures that you're looking at are pieces of technology that were developed through scientific knowledge. Okay, again, okay, the application of scientific knowledge. Technology changes over time because it's based on discoveries that have been made in the past. So as technology becomes more advanced, new information can be discovered. New discoveries may refute previously accepted theories and ideas, causing new experiments to be performed, new data to be gathered, new theories to be formed about older ideas, and new technology is then developed. Okay, so that is the end of your first lecture.